Okay, class, we're going to start talking about implantation. Implantation. That is when the blastocyst enters into and burrows in the endometrium stratum functionale of the uterine wall. So let's go back for a moment. Let's review. We started out with sperm meeting egg, fertilization zygote. We went from here, and what you don't see is a two-cell stage. And each cell now in this terminology is called a blastomere. The cells are cells, but they're called a blastomere. So we go from this to a two-cell stage to a four-cell stage. This is still traveling down. These cells are contained still in the zona pellucida. By doing that, the cells will do, and now we'll go back to cell divisional cycle. The cells multiply, but they don't get any larger. So they spend very little time in G1 of the cell divisional cycle. The reason being, if they were to get larger, they would block into this tube known as an ectopic pregnancy, or tubal pregnancy. Okay, so 4 cells, then a bunch of cells, about 16 to 32, that would be called the marla. It's like a bunch of grapes, the marla. And then, now we're going to go, as usual, migration and differentiation. The migration will cause some of the cells to migrate here that are going to be called the inner cell mass, or some authors call it the embryonic disc. These cells here is where the person develops from. These other cells here, differentiation, migrate and become what we call trophoblastic cells right here. Trophoblastic cells, extraembryonic cells. These trophoblastic cells will be responsible for creating the extraembryonic structures, like the placenta, the chorion, the amnion, the allantois, yolk sac, stuff like that. So again, here's the inner cell mass, trophoblastic cells. Now let's go to time-wise. About 4 days, let's say, this ball of cells, the blastocyst, has entered into the uterus. Now what it's going to do, in about 6 to 7 days, it's going to start to burrow into the wall of the uterus at the layer of the stratum functionale of the endometrium. So over here you can see on the 6th day after fertilization, the trophoblast cells secrete an enzyme that allows the blastocyst to eat into the endometrium. It thus burrows into the stratum functionale endometrium with the inner cell mass of the blastocyst going first. So this layer goes first. That's going to be the first one going in. By the 7th and 10th day, the blastocyst is completely buried over, as we'll see later. About 75% of pregnancies do not implant, thus have failed. And so it would just don't implant, go straight out. Lady would not even know she's pregnant, and really would pretty much have a menstrual period. Okay, so we go further. Again, here we are. Begin 6 to 7 days after ovulation with the trophoblast adhered to properly prepared endometrium. The trophoblasts then proliferate and form 2 distinct layers, do mitosis and form 2 distinct layers. One layer is called the cytotrophoblast, which is totally cellular, and the other is a syncytiotrophoblast, which is a bunch of cells that are fused together. So let's look here. The implanted blastocyst is covered over by the endometrial cells, and once that happens, about the 14th day, implantation has completed itself. So right here you can see, and I'm going to come over here too. Right here you can see, here would be the inner cell mass, trophoblastic cells, blastocyst cavity, also some call it blastocele. It is starting to want to burrow into the endometrial cells. Over here you can see that happening too. When that occurs, then some of these trophoblastic cells, proliferation from the cytotrophoblast, and the ones further from the syncytial. See that syncytial, which means a group of cells, but they don't have a cytoplasm because they're all combined. Okay. Lumen of uterus, blastocele, inner cell mass, what you see here. Okay. All right. So now we go a little further, and I'm going to go over here too. Okay. So now we're looking at the viability of the corpus luteum. Now let's remember, the corpus luteum was that developed after ovulation. So this is in the ovary. I want to keep the corpus luteum functioning. I want to keep it functioning, okay? Because I want the corpus luteum to continue to secrete progesterone and estrogen, which is necessary for the continuation of the pregnancy. The problem is the LH secretion from the anterior pituitary is way down. It's pretty much way down. So it could not maintain the corpus luteum. And if it cannot maintain the corpus luteum, then estrogen and progesterone cannot be produced and the pregnancy will be lost. The pregnancy will be lost. Okay? That's what they call an inadequate corpus luteum. Okay? All right. Or called the corpus luteum of pregnancy. Okay? So the trophoblastic cells themselves secrete an alternative to LH that's going to work like LH, luteinizing hormone. And that's called human chorionic gonadotrophin. So human chorionic gonadotrophin will then keep the corpus luteum going. The corpus luteum then can secrete progesterone and estrogen to keep the pregnancy going. Okay, so chorionic gonadotrophin, even before the 6th implantation, the trophoblast cells of the blastocysts release human chorionic gonadotrophin. It acts like LH, luteinizing hormone, to keep the corpus luteum alive to continue releasing estrogen and progesterone. Keeps the endometrium thick and vascular. That's another thing. To keep to house the implanted blastocysts, and it prevents menstruation. Secretion of HCG declines about the 10th week, but by that time the placenta has developed, and the placenta is going to take over the job of secreting estrogen and progesterone. So no longer will you need the corpus luteum to secrete the estrogen and progesterone because the placenta would have formed. So this corpus luteum and this HCG is just temporary until the placenta develops. Now, note this. See, the HCG is being produced by the blastocyst itself, the trophoblastic cells. So this is the substance. When a lady does the pregnancy test, here we go now. When a lady does the pregnancy test, she's looking to see if this is present. If this is present, she is pregnant. Now, I got to point this out. The pregnancy could be a normal one or it could be an abnormal. She could have what we call a molar pregnancy. A molar pregnancy, hydatidiform mole, is basically that the woman is pregnant, she's producing trophoblastic tissue but no inner cell mass. So it's really a tumor type of thing. However, we know that if the level is extremely high at a certain stage of this, that is probably a molar pregnancy and not a true pregnancy. But however, again, this is the substance that is measured to see if a lady is pregnant. And you can see they can do it as early as 6 days, particularly if it's done from the serum versus the urine. The urine, it doesn't appear until a little later. Okay? So we go a little further now. HCG, as we mentioned, the chorion develops from the trophoblast after implantation. So the chorion, we're going to change the name continues this hormonal stimulation. Between the second and third month, the placenta assumes all these roles. Okay, so let's go here. So you can see human chorionic gonadotrophin shoots way up. Estrogen progesterone is up, but it comes way down because it's not needed. It's not needed because the placenta will eventually take over. So that's what we're looking at. Okay. The formation of the placenta occurs from the embryonic trophoblastic tissues and maternal endometrial tissues, which basically means the placenta is part of the tissue of the placenta is from the mom, and part of the tissue is from the blastocyst, the developing new entity. Okay, we'll come over here, see what we have over here. Okay, again, that's the same type of there. Okay. The chorionic membranes, from day 7 to 12, the chorion splits into a 2-cell layer structure, the cytotrophoblast and schizotrophoblasts, because again, the trophoblastic cells are the ones that's making the chorion, so we can call it also chorionic. So I'm going to go back right here. You see this here, which is trophoblastic, can also be called chorionic. Chorionic. Okay. All right, we go further. So you can see terminology can change, and there are several terms for the same thing sometimes in this field. Okay. So I come back here. All right. Here we are. Okay. The chorion develops finger-like villi, which become vascularized, extend to the embryo as umbilical arteries and veins, lie immersed in maternal blood. The Deciduous basalis, part of the endometrium that lies between the chorionic villi and the stratum basalis. Okay, let's see if I have a picture. Sure, if I do. Okay, here is the okay. This right here, the covering of the endometrium around the implanted is called the Deciduous capsularis, which you don't see. The one back here of the trophoblast, the chorion between that and the mother stratum functionalis, we call the Deciduous basalis. Okay, so that's what we're looking at. So the decidual tissue is the mother's endometrial tissue. Okay, I'm going to come back. Okay, decidual cap partly surround the uterine cavity. Placenta is fully functional by the 3rd month. Okay, I'm going to come here. Now, let me come back here. Okay, and I'm going to go one more. All right, so we have Plastician, deciduous, the mother's tissue. Chorionic membranes, okay, at the same time the trophoblast is forming 2 layers, the inner cell fetal part forming 2 layers. You also have the amnion forming, formed by 2 germ layers. Now we're going to talk about germ layers. That's your ectoderm, mesoderm, and endoderm. Okay, so we'll come to that. But we also have the amniotic sac that's being developed. And in this drawing here, if I can find it, here's development of that amnion. So again, here's chorion. Matter of fact, let's just name some things. Here's chorion here. This part of the endometrium that's covering this whole thing is called the Deciduous capsularis. Okay. The portion out here, outside this, would be the Deciduous basalis. And because deciduous is the mother's tissue, and this is mother's tissue here. All right. But now I'm starting to get an amniotic cavity to develop that we'll come to again over here too. So just kind of concentrate on this. Let's not get too far ahead. Okay, so we're going to close right here.